Hey y'all, welcome to the last lecture of this unit. Today we're going to discuss the roles of cellular form and function. We started sort of working our way up these different hierarchies of organizations. So the cell is the basic unit of an organism. So all living organisms must be composed of cells. And they carry out the structural and functional properties of a living organism. Now we'll talk about higher organization in a couple later lectures, but right now you should just know that they're the ones at the end of the day that are responsible for all anatomical and physiological function. It's important to understand how the body works, how these cells can go rogue, and we get pathophysiologies, and then how we can target different therapies to help alleviate disease. So here are your learning outcomes for this first part. Cytology is the study of cells. Anytime you see ology, that's a study, and then the prefix cyt means cell. This really started when Robert Hooke first coined the word cellulae. And that was when he was looking at a cork and he saw these honeycomb-like structures, which the empty cell walls of the cork in the early 17th century under his crude microscope. Theodor Schwann, about 2 centuries later, hypothesized that all animals are made of cells. And then Pasteur was actually one of the first to demonstrate that in the mid-1800s, that cells arise from other cells. So life gives rise to life. Um, Around this time in the mid-1850s, there was this idea called spontaneous generation. And it was just that living things can sort of appear out of nothing. And he was the one that first demonstrated that this is not true, that cells must arise from some other precursor cells. So not that long ago, right? Less than 200 years that we've discovered the cell, and you can see all the advancements that have been made in medicine since. Cell theory encompasses a couple key aspects, and you should know how this developed. First of all, it's that all organisms are composed of cells and the products of that cells, which makes sense, and that you can't reduce the functional unit of the organism any simpler. So it's the simplest structural and functional unit, and any structure and function that the organism itself takes on are due to the micro activities of these cells. So for example, if I move a muscle in my arm, yes, that muscle is moving and the tendons are working, but at the end of the day, it's those individual muscle fibers that are contracting, causing that. Obviously, life comes from life, so cells only come from pre-existing cells through a process of mitosis or meiosis. And then they all exhibit biochemical similarities, which means that they use certain conserved mechanisms to carry out the same jobs. There are about 200 different types of cells in the human body, and their shapes are different ways that we can classify them. And you'll form, throughout your lab studies, you'll form up and look at some of these different types of cells under the microscope, different tissue sections. So I'm not going to belabor this point too much in the lecture. Just know that you'll be seeing those questions on the lab practical asking you to identify the types of cells. Let's review them real quickly though. Squamous is squashed, if you think about it that way, so it's thin, flat, scaly cells. Cuboidals are in the shape of a cube, which are square. Columnar look like columns, so they're taller than they are wide. Polygonal cells are irregular, angular shapes with multiple sides. Just a point of reference, this is very rare in normal anatomy and physiology. Cells tend to take on these rounded shapes at the corners, so seeing these angular shapes is rare. Stellate cells look like stella or stars. Spheroid or ovoid or round and oval. Discoid like a red blood cell or disc-shaped. Fusiform is like a muscle cell. It gets thick in the middle and then it sort of tapers off at the ends. And then fibrous look like a thread. An important thing to note is a cell shape can appear entirely different if you view it in a different type of section. So a longitudinal versus a cross section. So here's just some pictures to aid in your study of those different types of cells, and you'll be coming across some microscopic images in your lab exercises for you to identify. A good thing for you to be able to identify for lecture questions is an example of each type of these. I may ask you, give an example of a fusiform cell or a spheroidal cell or something like that. Most human cells average about 10 to 15 microns, but obviously there's a lot of variation there. Egg cells, you can actually see with the naked eye, they're 100 microns in diameter, so if you look very carefully, you can see an egg cell. And then some nerve cells are very thin, but they're over a meter long. So there's a lot of variation in this type. Um, the thing that limits usually cell size is physics, and that's if it's too large to support itself. So it's easier to have two small cells than one large cell. Obviously, if it's too large and it can't support itself, it could rupture. For a given increase in diameter, volume increases more than surface area. So these are just simple mathematics. You don't have to memorize this. That just explains why the larger you get, your volume increases more than your surface area, and your surface area is what's interacting with other cells, proteins, and the like. So that's why it's just not feasible to have cells that are overly huge. Please don't worry about memorizing this math here. I'm not going to ask you to calculate this. Just what I want you to appreciate here is when we double the diameter, we're really increasing the volume from 1,000 to 8,000. So this is why the bigger a cell gets, the less surface area to volume ratio you have, and that is that's the limiting determinant of how big a cell can get. So light microscopes are used most frequently, and you'll see those. If you're on campus and you're in the lab, you'll see light microscopes. And you can reveal the very basic components of a cell. Usually it's plasma membrane, its nucleus, and its cytoplasm. And then you have to have something like an electron microscope to improve resolution and see more details. So without getting too in the weeds of the physics, with electrons, I can hit that. So with a light microscope, you hit the cells with a beam of light. With an electron microscope, you hit them with a beam of electrons, and they give you a lot finer resolution. There are two types I want you to know about: transmission and scanning. So transmission electron microscope is the most high resolution. And what you see with a transmission electron microscope is a slice across the image. A scanning electron microscope, however, will give you a 3D surface feature, but it won't penetrate the cells. So if I want to see something on the surface of a cell, I use an SEM. If I want to take a section across a cell, I use a TEM. And here's an example of some of these. So this is with a light microscope, right? This is an eosinophil. And I can make out this plasma membrane here, and I can make out these nuclei, but really nothing else. It's harder to see. However, over here on this EM image, you see how the cell is sort of sliced out of your screen. I can see where the nucleus is, and I can also see the lysosomes and here. Fun fact for you here, this image is actually pseudo-colored because at this small of a size, color does not exist. So with an electron microscope, all you get is black and white images. And we have to pseudo-color those to get them to make sense. So there's a little fun nerdy fact for you there. Let's start breaking down the components of a cell, seeing what they're made of, and how those will eventually lead us to higher structures. So the plasma cell membrane is the coat surrounding the cell, and it defines its boundaries. So if you see 3 or 4 cells on a slide, that's what you're looking at on the outside is the plasma membrane. And the plasma membrane is a lipid bilayer, and there are also proteins that are stuck in that bilayer. The liquid contained inside the plasma membrane is the cytoplasm. So cyto meaning cell, and then plasm is the liquid within that. And the cytoplasm contains a bunch of different things. It contains organelles, the cytoskeleton, which helps give the cell its structure and its shape, inclusions, which are stored or foreign particles that are sort of granulated, and then the cytosol. Now, there's a difference between cytoplasm and cytosol. And some textbooks do not make this distinction, but I want you to know the difference. The cytosol is just the fluid, okay? The cytoplasm is everything in it. So it's the fluid plus the organelle, cytoskeleton, and whatnot. So make sure you can differentiate between those if you see them in a textbook or in another lecture. We call this the intracellular fluid, the cytosol the intracellular fluid, because it's inside, the cell. Then obviously we have the extracellular fluid, or the ECF, that includes what we call tissue fluid, or interstitial fluid. So ECF is everything outside the cell, ICF is inside the cell. So this is a rough structure of a representative cell, and it's a lot of detail here, but we're going to break these down one by one. For the lecture, I want you to be able to know definitions and functions. For the lab practical, I'll be asking you to identify these. So please don't worry about one for the other. Just know the definitions and the functional roles for the exam, and the identification will come later on the practical. So let's start on the outside, and then we'll work our way into some of the important organelles. So like I said, the plasma membrane is the border of the cell, and it appears as this pair of dark parallel lines when you view it with an electron microscope. It's because it's a bilayer, so you have two layers of lipids that orient themselves and provide you a double protection across the cell. So you have an intracellular face and an extracellular face of lipids. This will define the boundary of the cell, but it also governs the interactions with other cells because it has to cross by the plasma membrane first. It also controls what comes in and out of the cell, which is very, very important for homeostasis and to keep that cell alive. So here's an example of an electron micrograph of the plasma membrane. So you can see here that these are two cells stuck together. Here's the first plasma membrane with its two layers here and here. And then you can see the plasma membrane of the second one here and here. And then this is the nuclear envelope in the nucleus. The nucleus also has a bit of a membrane surrounding it. And one thing, and I'll touch more on this later, one thing that defines human cells from bacteria or prokaryotes is that we have membrane-bound organelles. So all of these organelles that I'll be talking to you about, the nucleus, the mitochondria, etc., are defined by a membrane. So you get these really nice defined structures under a microscope. So here's what a plasma membrane would look like. It has these little hills and valleys. All those cells look completely round to us. There are different indentions, just like if you look at Earth from space, it looks perfectly round. But if you walk outside, you see all these hills and valleys. That's the same way a cell looks at this layer. These are these lipid bilayers that orient themselves this way, and you'll find multiple different things stuck within that bilayer. Different transport proteins and channels that will allow things in and out. Cholesterol, which intercalates here between these lipids and keeps the cell membrane fluid so that it can move. Peripheral proteins that bind to the outside and the inside of the cell respectively. The cytoskeleton hold things together and the like that. 98% of that membrane is made of lipid, so there are plenty of proteins that play key functions, but lipid is the main component here. I like to think of the plasma membrane like I'm building a castle wall. 98% of that castle wall is going to be bricks, but every once in a while I'm going to have a gate or a window or a turret or something like that. So you can think of those lipids as the bricks of the wall. Most of these lipids will be what we call phospholipids, and they're amphipathic. And if you remember from your earlier lectures, amphipathic means that some part of the molecule likes water and some part of it doesn't. So you remember from earlier that there's intracellular and extracellular fluid. So it would make sense that the hydrophilic phosphate heads of these phospholipids orient themselves outward because they like water. Then on the inside, their hydrophobic tails will interact because those hydrophobic tails don't like water. They will spontaneously arrange like this so that they can give you that nice bilayer we were talking about. They will drift laterally, meaning that the membrane is constantly fluid, so it's not like a wall where one brick stays in the same place. You can think of it more like a bubble around the cell. Cholesterol is the rest of the lipids, and their job is to hold the phospholipids still, and they can alter the stiffness of the membrane based on heat and different things. And then there are also glycolipids, and glycolipids are phospholipids that have these little carbohydrate tags on their extracellular face. And you can think of these as sort of a cellular badge. So different cells can recognize each other based on the different type of glycolipids that they have. They also contribute to the glycocalyx, which is this sort of sticky coating on the cell surface that allows the cell to bind to different things and again to interact and recognize each other. Interestingly enough, membrane proteins are 2% of the molecules, but 50% of the weight. So these are very dense proteins that come in various shapes and sizes. Integral proteins mean that they penetrate the membrane, so they integrate within the membrane. There are several different types. Transmembrane proteins are just what they sound like. They're trans, so they cross the membrane. They pass completely through. And these are ways to connect the cytoplasm and the extracellular fluid. Obviously, just like lipids, the hydrophilic regions of the protein will contact water, while the hydrophobic regions will bury themselves into the lipids of the membrane. Some tend to drift and others are anchored. It just depends on the type. You also have peripheral proteins, which are only on one face of the membrane and do not penetrate it. They're usually tethered to the cytoskeleton to keep them from floating away in the membrane. So here's two examples of transmembrane proteins. You can see how it penetrates the phospholipid bilayer and is anchored to the cytoskeleton. These hydrophobic regions will be buried here within the lipids, and then the hydrophilic regions will be exposed to the water on either side of the membrane. Okay. So these have many, many different functions. If any cell communicates with the outside, it's through one of these proteins. So they can be receptors, second messenger systems, enzymes, channels, carriers, just about anything. If you can think of a cellular function, odds are it's carried out by a membrane protein. So here's some that I want you to key in on. These will be our key examples here. Receptors are proteins that bind chemical messengers like hormones. So let's say we have a hormone floating over here in the tissue in the bloodstream, and we want it to penetrate the cell. It'll bind to this receptor, and this receptor will kick off a signaling cascade inside the cell. Now, receptors are advantageous for a couple different reasons. Imagine somebody knocked on your door at your house. Well, you would want to know who that person was and if they were safe before you let them in, right? So that's what the cell does with receptors. You don't want to just let things across the cellular membrane because they could be toxic. So you want these different gatekeeper proteins. That's what receptors do. They bind things on the outside and then they signal to the inside if it's safe to let it through or if it's not safe to kick off some signaling mechanism here. Some are enzymes that break down chemical messengers and terminate its different effects. Some are channels, so when they see that a particular ion, like sodium for example, or potassium, is fit to come through, they open up this channel here and open up a small hole in the plasma membrane and allow the cell to exchange solutes with its environment. You can also have gated channels that are not constitutively open but only allow things during certain times. Cell identity markers, these tend to be glycoproteins that I told you were like little badges for the cells. And then CAMs. CAMs are cell adhesion molecules, and their specific function is to bind to other cells, to recognize each other, transfer information, or to bind each other to form higher order structures like tissues and organs. So this is just some more text. If you've noticed, whenever I have these online lectures, the slides are very text heavy. That's so you can go through and study a little easier without having to rewatch. So you can go through these on your own if you want to rehash what I just said. I do want to pull a couple things out of here. The different types of channel proteins. There are different ligands that can gate those. So ligand gated channels respond to chemical messengers. Voltage gated, like in your muscles and your neurons, respond to a change in the charge. So if they sense a change in charge, they'll open or close. And then mechanically gated changes, like in your ear that keep you balanced, respond to physical or mechanical stress on a cell. Okay. The other thing to note about some of these carriers is that these pumps are carriers that consume ATP. So it does take energy to pump something against its concentration gradient. I briefly mentioned second messengers, so let's go through an example of how that works because sometimes these signaling pathways can get a little complicated. So let's look at these in a little more detail together. So let's take one, for example, epinephrine. So epinephrine's floating around, and it's what we call a first messenger because it's the one that binds to a receptor. So a first messenger is the one who actively binds to the receptor. A receptor in the case of epinephrine, this isn't true for all receptors, this is just for this example, will activate something called a G protein. And a G protein is just something that gets its energy from GTP. And you don't have to know that detail. And then a G protein is going to relay a signal to a molecule called adenylate cyclase, which will convert ATP to cyclic AMP, but the big thing is it converts to a second messenger. And then that CAMP will activate cytoplasmic kinases. Kinases are enzymes that will allow some to turn on and turn off. And this is how a drug, for example, can work through a G protein and a second messenger. Let me tell you a little story to see if I can make this make sense. So you have your castle wall, right? And you've got a little guy sitting on top of your castle wall, and his job is to look out for messengers. So a guy comes riding up on a horse, right? Game of Thrones, Lord of the Rings, whichever one you want to imagine. Guy comes riding up on a horse, and he can't get through the castle, right? So he yells up to that guy on the top of the wall, and he says, "You got to let him know that there are enemies on the way." So what is the guy on the wall going to do? He's going to check the signal from outside, and then he's going to go running through the town. And he may go and tell the king so he can prepare the army. He may go and tell the blacksmith so he can start making, some tools and some weapons. He may tell, I don't know, other members in the town to go seek shelter. And that one signal from outside causes multiple different signals inside the town so that they can prepare for whatever that stimulus is. This is exactly how the body works to take one signal outside of the cell and transduce it to many other signals inside the cell. And we call that signal transduction. So it's not the case that one molecule signaling causes one other molecule to signal. It's typically in a cell, one molecule can cause 10, 50, 100, 200, 1,000 other molecules to signal. So this is an extraordinary complex process which we call signal transduction, and that's how second messengers work. So here's that example just in a graphical format. A first messenger binds the receptor, kicks off to make a second messenger, and then goes through and activates other enzymes and such like that. You do not have to memorize this specific GTP or G protein coupled receptor mechanism. I'll give you something on the exam, probably that you've never seen before, but you should just be able to identify what the primary and the secondary messenger is based on those concepts. All right, so that's enough on the plasma membrane. Let's look at the glycocalyx. So the glycocalyx is that fuzzy, sugary coat that is external and guarding the plasma membrane. Different carbohydrates here of glycoproteins and glycolipids. And interestingly enough, each individual has a unique glycocalyx except for identical twins. So this tells us that the structure of the glycocalyx is actually governed by our genetics and by our DNA. Multiple different functions here, obviously protection and immunity, defends against cancer. Transplant compatibility has to do with glycocalyx matching, cell adhesion and stickiness, fertilization, as well as embryonic development. There are also these little substances called microvilli, and they're these little fingers that extend from the membrane. And what they do is their main function is to increase the surface area of the cell. So in cells in the intestines that are specialized in absorption, you see a lot of these because the more surface area, the more efficiently you can absorb, right? So imagine them as being the tiny little threads on carpet, reaching out and increasing surface area. On some absorptive cells, they're very dense, and we call that a brush border. And some of them have these actin filaments that sort of milk whatever they absorb into the cell. This is what those little microvilli look like. See, they look like little fingers. And if you cross-section them, you can actually see those little actin microfilaments. This is one microvillus right here, or right here, for example. And you can see those actin microfilaments that make up the root, if you will, of those. All right, don't get tripped up between cilia and microvilli. So microvilli are only 1 to 2 microns. Cilia are a lot larger. They're 7 to 10 microns. And there is a single non-motile, what we call a primary cilium, found on almost every cell. And it's like an antenna, right? And what it does is it monitors nearby conditions. There are some in your inner ear that are sensitive to balance. There are some in your retina that are sensitive to light detection, but almost every cell has these. And then you have multiple non-motile cilia, and those are found on things like the sensory cells of the nose. Ciliopathies are what we call diseases in structure and function of the cilia, so different congestive disorders. Motile cilia are found in places like the respiratory tract, the uterine tubes, the brain, and the testes, and their job is to keep things moving. So they actually beat in these waves, and just like waves moves things, wash them ashore, it will sweep things in one direction on a power stroke and then a recovery stroke. So for example, when you're coughing something up in your lungs, that's the result of what's called the ciliary escalator, and those cilia in your respiratory tract beat upwards. And they beat all that mucus, which traps dust and pathogens and stuff like that, allowing you to cough it up and keep it out of your lungs. The axonomy is the core of the nodal cilium, and this is very important. You'll see this. It has a 9+2 structure of microtubules, which means you have 2 central microtubules surrounded by a ring of 9 pairs. And that anchors the cilium as part of the basal body, and these different molecules can crawl up the microtubules and bend it so you can move it as needed. And obviously, it's important to remember that all these cellular processes require ATP, require a lot of energy. So here's that 9+2 arrangement I was showing you about. Here's a real-life electron micrograph of that. 2 here in the central ones, 9 pairs, so 18 total of the peripherals. And you can see here how they look like little anemone. See, I can't even say that. It's like Finding Nemo. Anemone that can beat in one direction or the other and keep mucus going. So cilia will normally beat freely with a layer of saline at the cell surface. This has to do with the chloride pump that pumps chloride into the ECF and allows for the expulsion of sodium and water, and that allows this mucus layer to focus on top of saline. Just a quick little tidbit for you. This explains the pathology of individuals with cystic fibrosis. Individuals with cystic fibrosis have a mutation in the gene for this chloride pump. So because they can't pump chloride correctly, their cilia can't beat correctly, and they get that thick mucus caught up in their lungs that they can't cough up. So that's an example of a disease that we see at the organism level that goes all the way down to the molecular level with the movement of chloride ions, just so you can keep in context the role of the hierarchies of a complication. This is just to give you an example of how those cilia will beat and keep that mucus layer moving. See how they'll beat down, and when they recover, they sort of whip backwards. So they're not like windshield wipers where they go back and forth. They'll sort of beat and then like work themselves up kind of like a snake, if you will. This is that slide on cystic fibrosis. I forgot to put the slide in here. I didn't want to miss it. Like I said, it's hereditary, in which you don't have the correct installation of the chloride pumps, and then you don't have the right saline layer. You get mucus plugs on things like the pancreatic ducts and the respiratory tract, and this obviously can lead to early death due to chronic respiratory infections. Right? So flagella are sort of cellular tails. The only type of cell in a human that has a functional flagellum is a sperm. And you've seen the image of a sperm with its little tadpole kind of structure. And it looks like a whip with something called an axonomy that's identical to a cilium's, but it's much longer and it's stiffer. So you get this snake-like sort of corkscrew motion, so you don't require a power stroke and recovery stroke, which means you get very fast swimming movement in the opposite direction here. Pseudopods are really cool. When you look at a picture of an amoeba, that's what you see is pseudopods. And they're these little finger-like projections of the cell that vary in shape and size, and it kind of looks like a cell is crawling along. And you can use these for capturing foreign particles or for cellular locomotion as well. See how those pseudopods will sort of reach out here like little finger-like projections, and then reach out and engulf different substances. So let's discuss a little bit of membrane transport. So the plasma membrane is selectively permeable, meaning we want to allow some things to come through, but we don't want others to come through. So for example, I want my family to be able to come through my house, but I don't want a bear to be able to come through my house. So I have to be able to recognize who's at my door, the family member or the bear, right? So plasma membrane integrity is very important to the survival of the cell. There are passive mechanisms that do not require energy and active mechanisms that do require energy. So passive mechanisms are usually things that are based on water movement, so like diffusion, filtration, and osmosis. Active mechanisms are things like active transport and vesicular transport. And then you also have carrier-mediated mechanisms, which is where a protein itself does the work of transporting substances across the membrane. Let's go through these one by one. Filtration is where you drive something through by pressure, okay? So it's not an osmotic gradient, it's a pressure gradient. So for example, water and small solutes through your capillaries in the endothelium of your blood allow you to deliver water and nutrients to tissues, and it's also the mechanism by which your kidneys filter waste. So this has to do with blood pressure, interstitial pressure, pressure gradients driving something in or out of a cell. Okay? So here's an example of filtration through the wall of a blood capillary. These are the endothelial cells that make up your capillaries, and you can see here that there are holes and clefts in between there, but there are some things, small things, that are able to escape. Simple diffusion is when particles move from high concentration to low concentration, and this is due to constant spontaneous molecular motion. When molecules are moving together, they will collide and bounce off, and there's energy in those collisions, so that can drive diffusion. Remember that just like a ball rolling down a hill, substances diffuse down their concentration gradient. So this doesn't require necessarily a membrane. The substance can diffuse just if it's permeable to that substance. So membranes are permeable to some things and not. There are some general rules that I want you to know that are trends that affect diffusion rates through a membrane. So for an example, if you have a temperature that's higher, you have more motion, so it'll increase diffusion, right? So what you should be able to do with these different variables is if I give you a set of parameters on the exam, 2 sets of parameters, you should tell me which one will favor diffusion higher. Molecular weight, obviously large molecules move slower, so glucose, for example, would move slower than salt would. The steepness of the concentration gradient, right? So if there's a big difference, I'll increase the rate. So if I have a higher hill, the ball will roll faster, right? So the higher that gradient is, the more I'll diffuse. Membrane surface area. If I'm trying to get into a football stadium and there's only one gate, I'm not going to have a lot of diffusion. But if there's 20 gates, I can move people real fast, right? And then membrane permeability. Can my substance of interest actually get across the membrane? Osmosis has to do with how water moves things, and certain solutes will just follow water along. Water also goes toward its concentration gradient, so a place of water, water will move from areas of high water concentration to low water concentration. But what this means, make sure you don't get this mixed up, is that water will flow from low solute concentration to high solute concentration, right? Because water and solute are in opposite directions. So make sure you don't get mixed up on the exam. Water follows solute. So water will move to where it can be less concentrated down that concentration gradient. So particles like sodium, an example, for one, we refer to that as drawing water. So water will follow these solutes. This is a big consideration for IV fluids, and partially why we put different ions like sodium, potassium, etc. in IVs. We have to have osmotic balance to treat things like diarrhea, constipation, edema, etc. Now remember that water can diffuse through a phospholipid, but there are special proteins called aquaporins, which are just what there are. They're pores that transport water through the membrane to speed it up a little bit if they need to. A cell can make more aquaporins to speed up osmosis. So, for example, let's say I have a membrane here and I have more solute over here. Over time, water will go over here to be with that solute. So that solute is known as what we call drawing out. So this will exert a higher osmotic pressure, right? Because there's more stuff over here. That's osmotic pressure. And then less hydrostatic pressure. So osmotic pressure is the hydrostatic pressure required to stop osmosis. So it increases as your solute rises, and then you can actually override this through a process called reverse osmosis. And that's where we push down on it enough to where water is forced out the other side. And this is actually one of the ways that we purify water for drinking. So what is osmolarity as a concept? So 1 osmole is 1 mole of dissolved particles. So it takes into an account whether the solute ionizes in water. So for example, 1 molar glucose is 1 osmolar because glucose cannot ionize, right? But for example, 1 molar sodium chloride ionizes into Na plus and Cl minus. So that gives us 2 osmolar. So remember, your take-home message for osmolarity is how many particles do we get. So if we did calcium chloride, for example, CaCl2, that would be 3 osmolar because of 1 calcium and 2 chlorides. So work through those in a couple of practice problems to make sure you can make that make sense. Osmolarity is the number of osmoles per liter of solution. Obviously, body fluids contain a mix of many chemicals, so the osmolarity is the total concentration of all those particles in your body fluids. In general, and this can change during many states, plasma, tissue fluid, and intracellular fluid are about 300 milliosmolar. So that's a good number for you to know, especially in the clinic. So this brings us to the discussion of tonicity. And tonicity is just what it sounds like, it's tone. And that's the ability of a surrounding solution to affect the fluid volume, depending on concentration. So for example, if we had a hypotonic solution, it will cause a cell, and we drop a cell in a hypotonic solution, it will cause a cell to absorb water and to swell. What this means is that a hypotonic solution has a lower concentration of solutes than intracellular fluid, so distilled water. So if I give a patient a huge amount of distilled water, it will cause their cells to swell because the cells are saltier than the water is, right? A hypertonic solution causes a cell to lose water and to shrivel or crenate. So for example, if I gave a patient ocean water, the ocean water is saltier than the cells, so they will shrink, right? Does that make sense? Water will flow from areas of low solute to high solute, so it'll go from the cell to the extracellular fluid. Isotonic solution, like normal saline, causes no change in the cell volume. So this is what we're shooting for, right? We don't really want to give patients hypotonic or hypertonic solutions unless their particular pathology calls for it. We want to give them isotonic solutions so we're not altering cell volume, so that the concentration is the same, like normal saline or lactated ringers, for example. And here's what we look like an image of tonicity. So isotonic means this blood cell looks just like it's supposed to. It looks like a biconcave disc. Hypotonic, you see where the cell has swollen. So it's like a beach ball. We've overinflated it. With hypertonic, we've sucked water out of it because it's saltier out here than it is here, and we have crenated it. So we've made it shrink up. Okay. So that's all the passive mechanisms. Now let's go on to carrier-mediated transport. So transport proteins, like I told you before, can carry things in or out of the cell or out of an organelle since they have membranes too. Carrier-mediated transport is much more specific. So they're specific for particular solutes. The solute will bind to the receptor on one site. The receptor will change shape and spit it out on the other side. Very simple. However, transport proteins can become saturated. And that's at a point where there are too many molecules that all my transport proteins are full. We call this the TM, or the transport maximum. And that's where all my carriers are running as quick as they can. So for example, think about like UPS during Christmas time. There's only so many employees that UPS have. So they're processing as many packages as they can. That may be why your package gets delayed. So that's the transport maximum. Everybody's transporting at the maximum rate they can, but some things could get left out. So this is the example of how that transport maximum curve will look. 3 types of carriers you should know about: uniport, symport, and antiport. Uniport carries one type of solute. So for example, calcium pumps are specific for calcium, nothing else. Symport carries two solutes simultaneously in the same direction. Sometimes you'll hear this called co-transport. So for example, the sodium glucose, the sodium glucose transporter. Finally, you have antiport. An antiport carries two or more solutes in opposite directions or counter-transport. So for example, this is the sodium-potassium pump, which removes sodium and brings in potassium. Now there are three mechanisms to do this transport, and they're facilitated diffusion, primary and secondary active transport. So let's go through those three. And just a little side note here, I would make yourself a flow chart of these so that you can classify and understand all these different types of mediated transport and get them sort of together in your brain. So I would highly suggest that in your lecture notes once you're done with the lecture. Facilitated diffusion is when the carrier moves down its concentration gradient. So it is diffusion, it doesn't require energy, it's just going from high to low, it's running down the hill, right? You'll attach to the carrier will change conformation and release it on the other side. Just spit it in from one side of the cell to the other, okay? So for example, this is extracellular fluid. The solute will bind to the receptor will check it, and then it will spit it out on the other side. Very simple. Right? Now we get to things that are expensive that require ATP. Primary active transport is when you're moving something against its concentration gradient. So this is where you move something in an unfavorable direction that requires energy to be consumed. The two most common examples of these that you need to know very, very well are the calcium pump and the sodium-potassium pump. The calcium pump is a uniport pump, so it uses ATP to expel calcium. And then the sodium-potassium pump uses ATP, but an antiport to expel sodium and import potassium into the cell. Sodium-potassium pump, you have to know this thing backwards and forwards for the exam because it's going to come up through the rest of your career. Sodium and potassium balance is basically one of the most essential ionic interactions for cellular life and for life and physiology in general, so that's why it's so important. We're not just teaching you this to torture you with a lot of information, it's vital. So each pump consumes one ATP, and it exchanges three sodium for two potassium. And what this does is it keeps potassium concentration higher in the cell and sodium lower in the cell. So a little way for you to remember this is it's salty outside the cell. So sodium and chloride are high outside the cell. Potassium is high inside the cell. The reason this is necessary is because the membrane is leaky between sodium and potassium. So you wouldn't be able to keep this concentration correct without it. So you're fighting this leak in the cell. Half of your daily calories are utilized to run this pump, which seems insane, right? But that should tell you how important this is for cellular function, okay? So this is what the sodium-potassium pump does. For every ATP it consumes, 3 sodium get spat out, 2 potassium get spent in, okay? So for an example, if I gave you a problem on the exam and I said, okay, well, your cell just consumed 6 ATP. How many sodium did I spit out? Well, 6 times 3 is 18, right? So you should know it well enough that you can manipulate it back and forth. A little mnemonic that helped me when I was learning this to keep the direction straight is 3-2-1 Nokia, okay? 3-2-1 Nokia. 3 NO, so sodium out, NO. 2 Ki, so potassium in, 2 potassium in. A, 1 is A, you use 1 ATP. Maybe that'll help you, maybe it won't, but hopefully that'll help you remember. 3-2-1 Nokia, 3 sodium out, 2 potassium in at the cost of 1 ATP. Okay? Know it cold. I guarantee you there are going to be a couple exam questions about this. So like I said, it maintains the concentration gradient, allowing for secondary active transport. What this does is drives other transport mechanisms, regulates your solute concentration and osmosis, make sure that the resting membrane is negatively charged, and it also produces heat. So you can see now why this thing eats up half of your calories, because it's very, very, very important for a lot of other processes. And what about secondary active transport? So this is an indirect usage of ATP, and I'm not going to test you on the details of this one as much as I am the primary active transport like the sodium potassium pump. But basically, is an indirect use of ATP. So it uses another protein's ATP usage to perform its function. Let me see if I can make that make sense for you. So the sodium glucose transporter is a symport transporter. It moves glucose into the cell, carrying sodium down its gradient. But it only works if the sodium potassium pump is working. So the sodium potassium pump is pumping sodium, and it is using ATP. But the sodium glucose transporter is not. It's using the sodium gradient. So pump 1 over here creates the sodium gradient using ATP. Pump 2 uses the sodium gradient without using ATP to transport glucose. So it's basically using the work of another device. Imagine you're driving a car down the road, right? You're the one using the gasoline, but if somebody hitches a ride on their bike and holds on to the car, they're being transported, but they're not using any energy, right? They're just riding along on your car's energy. So that's kind of how a secondary active transport works. For example, these work in kidney cells that have the pump at the other end of the cell, and this is the mechanism by which glucose is conserved in the urine unless you have diabetes or something like that. So this is secondary active transport here. Because we're creating this sodium gradient, we can harness it on this end to bring in glucose. Sodium is being pumped out, so there's net negative sodium in the cell. We take advantage of that by bringing it back in on this end, and we can bring glucose along with it, saving ourselves a little bit of energy. Vesicular transport is next. This is where we move very large particles, fluid droplets, or other molecules through the membrane in vesicles. And a vesicle is just like a bubble. Endocytosis is where we basically wrap the membrane around and just bring in a bubble of stuff. And there are different mechanisms specifically for this. Phagocytosis is one, cell eating. So things like macrophages, phagosomes, pseudopods will sort of just reach around and grab something and bring it in. Penocytosis is the same thing, except it samples extracellular fluid and whatever might be in that. So the membrane caves in and pinches off into a vesicle. And then you have something called receptor-mediated endocytosis. And that's where a particle will bind on a receptor, and the membrane will close around and digest the particle and the receptor together in something called a clathrin-coated pit or a clathrin-coated vesicle. And it will just sort of eat in the whole thing and bud it in to be processed. Exocytosis is the same thing. We send something from the center of the cell to the periphery in a bubble. That bubble merges with the membrane and spits it out. All of these, however, do require motor proteins energized by ATP. Let me see if I can make it make more sense here. So this is a phagocytic cell, right? So its job is to eat things. It'll form these pseudopods and eat this little particle here, and it'll sequester it in something called a phagosome. Now, a phagosome merges with something called a lysosome and becomes a phagolysosome. Makes sense, right? And then this phagolysosome will digest whatever particle it might be, foreign particle, and then spit out the waste products. So this is endocytosis up to here, and then spitting it out is exocytosis here. So you see how that bubble just kind of fuses with the membrane and then spits out whatever leftovers are there. So receptor-mediated endocytosis is obviously more specific, so you can take in specific molecules that bind to specific receptors. Clathrin-coated vesicles are the mechanism by which you uptake cholesterol from your bloodstream. So this is how LDL works in that. Here's a beautiful image on electron microscope of receptor-mediated endocytosis. You see here the LDL binding to the LDL receptor. It sort of pinches into a bubble, and then right here it'll close off to make the plasma membrane again, and then send that bubble inside the cell to be processed. Transcytosis is where you transport something on one side of the cell and release it on the other. So, like during digestion. Exocytosis is whenever you get rid of the junk that's left. So here's an example of transcytosis. Let me see if I can make this make sense for you. So this is the capillary endothelium here. This is the inside of a blood vessel. So this is how something will go from a blood vessel, get pinched in by the capillary, and release it here in the tissue fluid to perhaps a muscle cell, for example. So this is how those individual particles will cross over to get to where you need them. And then this is another example, one more time, of exocytosis, where you fuse and you release these things. You don't have to worry about the details on this slide. I just wanted to give you a couple pictures so you could picture that in your mind of how we're bringing things in the cell and taking them out. All right, so that is the outside of the cell. Let's talk about the inside briefly, and then we'll wrap up. So the cytoskeleton is what holds the cell together. It's a network of protein filaments and cylinders. It determines the shape of the cell, supports its structure, organizes its contents. Imagine it as the musculoskeletal system. Like that is to your body, helps you move, helps you stand straight. That's what the cytoskeleton is. And it's composed of a couple different filaments I want you to know about: microfilaments, intermediate fibers, and microtubules. So a lot of times when we see a picture of a cell, right, it's just this blob of everything floating around. Well, it's actually not really true. The cell's very, very organized, and it has this network, right, of these different filaments that act as different highways, so that we can move organelles, we can move different things across here. Don't memorize this figure, obviously. We'll talk about some of the important ones in detail. So let's talk about these different filaments. Microfilaments are the smallest. They're made of actin, and they form what's called the terminal web. The intermediate filaments are a little bit bigger, and they're, for example, made of things like keratin. And the intermediate job is to give the cells shape and resist stress. The microtubules are the biggest, and they're at 25 nanometers, and they're made of tubulin. They actually radiate from the middle, so they look like a 3D bike tire, for example. They have like the spokes. And they also hold the organelles and are important for transportation. So these motor proteins can walk along the microtubules and form the mitotic spindle and stuff like that. You should be able to rank these in order of their size and also to tell me what their main component is. So see, for example, these microtubules are made up of these small little units of tubulin, and they make these protein protofilaments. Organelles are the internal structures of the cell, and they carry out the specific metabolic tasks. So for example, if you have a company, you'll have different departments in the company. The cell is the company, and the departments are the organelles. So a characteristic of eukaryotic cells is that they have membranes. These are the membrane-bound the membrane-bound organelles. The non-membranous organelles are things like ribosomes, centrosomes, centrioles, and basal bodies. The nucleus is the largest, and usually it's the easiest thing to see on a microscope, right? It's usually the big, darkly stained dot in the middle. Most cells have one nucleus, but some things like skeletal muscle, for example, can have multiple nuclei. The nuclear envelope is another double membrane. So you have a double membrane around the cell, and then you have a double membrane around the nucleus that has different pores, just like the cell membrane does, to regulate what kind of traffic goes through the envelope. So remember, your nucleus contains your DNA. So that's why you have another double membrane and other nuclear pores, so you can really have tight regulation and control of what's coming in and out of your nucleus. The nuclear envelope is supported by the lamina, which is its own web of protein filaments. The nucleus, really, if you think about it, is like its own little cell within a cell. And it helps regulate the cell life cycle. It has its own cytoplasm called nucleoplasm, which is composed of chromatin, which is where your DNA is stored, and the nucleoli, and that's masses where the ribosomes are produced. So see, this is the nucleus, this is the nucleus here. This is the nucleolus, the nuclear envelope encasing it, and the nucleoplasm within it. And if you look on, so this is a TEM. This is a good point for me to go back and hop on that teaching moment from earlier. That's a good TEM image, see, because it's a flat section. You see how this SEM image is 3D and looks like you're looking at the surface. This is the 3D surface of the nucleus, and you can see those pores to get things in and out. Isn't that incredible? That's 1 micron. You can get some really cool pictures with SEM. So the nucleus is here. You have those nuclear pores, the chromatin that holds all your DNA and a meshwork around it. And then budding up to the nucleus are the ribosomes. And the ribosomes are important for protein synthesis. So the endoplasmic reticulum is these levels of channels and cisternae right by the nucleus. And the rough ER is where the ribosomes are. And this is the site of protein synthesis here. So remember, we go DNA, mRNA, protein. This is where the mRNA will be made into protein. It's continuous with the outer membrane because you want to be real close, right? mRNAs are very fragile, so that's why we want to be close here. It produces phospholipids and proteins of the plasma membrane, and it synthesizes proteins that are packaged in other organelles or secreted from the cell. The smooth ER has a little bit more of a fluid role. They're in the same network, but they function differently. It does not have ribosomes. So they're continuous with the rough ER, but they synthesize things like steroids, lipids, and they play a role in detoxification of alcohol and drugs and also store calcium. So they're not really synthesizing proteins. Their job is lipids and other molecules. So here's how you can tell the difference. For example, on your lab images, the rough ER is dotted with these little ribosomes. The smooth ER has none. Ribosomes are these little granules where the protein and RNA meet in something called tRNA, transfer RNA, and that's where that protein is made. They read the genetic messages, the mRNA, and then assemble amino acids in order specified by whatever codons are in that mRNA. So next we have the Golgi, and the Golgi, I mentioned UPS earlier. The Golgi is like the post office, right? So it's going to take things and it's going to package them correctly and send it somewhere. So it'll touch up any protein synthesis, stick a carbohydrate on it as a little tag, and send it off. Carbohydrate is like a stamp in this case. It receives these synthesized proteins, sorts them, splices it, adds carbohydrates, and packages them into vesicles. And we can ship these vesicles to different areas. They have what's called a signal sequence on them. And for example, this vesicle may be marked for the plasma membrane. So it'll shuttle it via motor protein over to the plasma membrane where it can do its job. So this is the Golgi complex here. It's kind of this loose budding structure that will take things, package them, and then send them off. So lysosomes are important because lysosomes are a package of enzymes that are bound by a membrane. These are generally round, but they can be highly variable in shape. And they can have these enzymes that degrade things. So they undergo hydrolytic digestion of things like proteins, nucleic acids, carbs, whatever it may be. A cell can use an interesting mechanism, this is really pretty cool, called autophagy, which is where if you have a defective organelle, instead of the cell undergoing apoptosis, you can just eat that organelle and reuse the cellular or the subcellular components of it to make a new one. So it saves the cell some energy. Autolysis is cell suicide. So if a cell can't function anymore properly, it can use a lysosome to eat itself from the inside out so it doesn't cause diseases to neighboring cells. Peroxisomes are similar, but they have different enzymes and they're produced by the ER. What they do is use molecular oxygen to poke holes in things. They produce hydrogen peroxide and they neutralize free radicals to detoxify things, but they can also play a role in host defense in your immune system. For example, you see a lot of these in the liver and the kidney where there's a lot of detoxification and blood filtering respectively. So here's some example of lysosomes and peroxisomes. They tend to be easy to find because they have those very nice circular structures. Proteasomes are also like lysosomes and peroxisomes, except their job is to break down proteins that are old and worn out. So the older your proteins get, you start tagging them with a molecule called ubiquitin. And after it gets so many tags of ubiquitin, the cell says, hey, we really need to get rid of this guy and build a new one up. So it'll send one after a proteasome after these proteins. They only degrade the tag proteins, right? Because you wouldn't want it to degrade healthy proteins. So it breaks it down into those peptides and amino acids, into those building blocks, so we can reuse it again, be more efficient. So see, you tag that protein, and then the proteasome will spit out the building blocks for use later. Mitochondria is the powerhouse of the cell, right? I'm sure you've heard that since first grade. Its job is to synthesize ATP. As I've discussed in context earlier, all of these cellular processes are very expensive. They require a lot of energy, and ATP is the currency for that. Mitochondria change their shape depending on their function, and they have a double membrane, just like the nucleus. There are spaces between those called the mitochondrial matrix. And it's interesting that mitochondria actually have their own DNA. And it's thought that original original cells did not have mitochondria, and they swallowed smaller cells that are now what we call mitochondria because they have their own DNA, they have their own mechanisms for energy production. So there's some pretty cool theories about how the mitochondria actually came to be. Um, Obviously, energy is extracted from organic molecules, transferred to ATP for storage, and then when we need it, we can use that ATP. Mitochondria are probably the coolest looking organism. They have all these little cristae here, which are those little folds and invaginations within the membrane, and then you have the inner membrane space, and then the outer membrane as well. This is what I was telling you about earlier about the evolution of mitochondria. It's really thought that they were once an independent species from bacteria that invaded primitive cells, survived, and they became kind of symbiotic, right? And the evidence for this is that they have circular cDNA of bacteria. They're inherited through the mother. It mutates more rapidly than nuclear DNA. So, for example, different metabolic disorders are usually due to mutations in mitochondrial DNA instead of genomic DNA. Centrioles are another important part. They're short assemblies of microtubules arranged in 9 groups of 3 each instead of the 9+2 rule like we talked about earlier. And they play an important role in cell division and also in cilia and flagella. So I don't want to go into the details of centrioles, but they play a role in movement here. There's an example of one there. Inclusions are not organelles. They're sort of just clumped up material. 2 types are endogenous and foreign. The endogenous ones are stored cellular products like glycogen, pigments, and fat droplets. Foreign things can be like viruses, intracellular bacteria, dust particles, and other debris. They're never enclosed within a unit membrane, and they're also not essential for cell survival. So that's the end of this lecture. If you have any questions, let me know. You're ready to go ahead and take the exam when it comes up. So if you have any questions, please let me know. And this will end Unit 1. Best of luck studying.